So hi everyone, thanks for being here. And I would be talking about my research project, which is the development of rapid high resolution spectroscopic imaging in the human brain. So here's a brief outline of my project. I would be talking about Alzheimer's disease because that's what my project deals with, and the need for early intervention, which could also help in um, development of techniques that can prevent the disease. Then I would be talking about the method I would be using in my project to approach this problem, which would be followed by my pulse sequence development, which involves a trajectory design, its implementation, and validation. Um, After that, I would be covering a summary of my current progress and the future milestones in the project. So, um, yeah, Alzheimer's disease is a neurodegenerative disease that slowly destroys memory and thinking skills. And this leads to like the person not being able to move, and they are not even able to carry out the simplest tasks out there. And Alzheimer's disease is the most prevalent cause of dementia among the senior population and is also one of the leading causes of death worldwide. Um, so here's the pathology of AD or Alzheimer's. It's characterized by amyloid plaques and neurofibrillary tangles. And the problem is that the diagnosis of Alzheimer's is only based on a marked cognitive decline that happens with age, but a confirmed diagnosis can only be carried out through a postmortem identification of these plaques. And also, we can see that in the case of Alzheimer's disease, the brain is significantly shrunk, and also there is a ventricle enlargement here. So, this is why this points out to the problem that the disease progression is so slow that we can only confirm it after the death of the patient, and we are not able to do anything while it's progressing. So this just draws attention towards the pressing need for early detection and intervention of the disease. So to do this, accumulating evidence has been pointing out towards the altered glucose metabolism in Alzheimer's pathology. So it's seen that the glucose metabolism is disrupted, and so this affects the production and clearance of the beta amyloid and tau phosphorylation, and this induces neurodegeneration because um, because we aren't able to supply the nutrients to the brain cells as it would be in case of a normal human being. So that's why there have been different methods that have been uh, in picture for quite some time now. So glucose metabolism has been used for many years as a biomarker for identifying regional pathology. And also there have been a lot of publications about FDG PET, using FDG PET as a gold standard to identify biomarkers. So, but there are some limitations around using FDG PET, and some of them include it has ionizing radiation and it's not suitable for pregnant women. And then it doesn't offer the flexibility that MRI offers, and also the tissue contrast. In terms of tissue contrast, things are very limited. So that's why my method would be talking about magnetic resonance spectroscopy. So magnetic resonance spectroscopy is just it's just a series of tests that are added to an MRI scan. And so this comes by imaging the water protons. And so in MR spectroscopy, um, we look at the metabolites that are located in a voxel, and so we can see that we can localize different metabolites, and so we can just focus on a region of interest. And so this is a spectrum that's obtained by the Fourier transform of the time-independent free induction decay, or the FID. And after the Fourier transform, if we do water removal, because water and fat are the major components of any voxel, so it's very important to remove them. So after we do that, we do notice that there are several metabolites here. And uh, now, interestingly, it's been found that in Alzheimer's, these metabolites are altered. And so investigating these alterations in metabolites offer immense potential for early detection of the disease. So to this end, my project aims at developing a rapid high-resolution spectroscopic imaging sequence. Now, this is because conventional MRI can be very slow, and so that's why it's not very optimal for patients to be sitting there for longer periods of time. So that's why the design criteria for my pulse sequence imaging sequence is that it should have a rapid data acquisition time, ideally less than 15 minutes. Um, and also at the same time, it would be offering high resolution, and also high sensitivity. This is achieved by using shorter echo times. I'll not go into the details of this because this is very physics related. And then the SNR, which is the signal to noise ratio, should be also pretty high, ideally. So that's why I would be using an approach which is called ROSETTE spectroscopic imaging. So the thing with rosettes are that they have high spatial resolution because they go far out in k-space. And so each trajectory goes like this. So there are circles that help span the entire k-space. And so because of this, they offer the relative ease of generating and implementing gradient forms. And since the gradients vary smoothly in time, this um, also kind of obeys all the limitations with regard to slew rate and gradient limitations. So this is the equation that I'm using in my sequence, in the development of my sequence. And so we first start with the k-space equation, and then so after we have the k-space data points, we calculate the gradient waveforms. And so the gradient waveforms are complex, so we obtain the real and imaginary parts. And so our focus is the real part of the like the real part of the gradient. And the constraints to the gradient design are that it's limited by the oscillating frequency or the rotational frequency. And there are also slew rate considerations because of the hardware limitations in the scanner. So in my case, I would be using the same rapid oscillation frequency and the slow rotational frequency. And this is because it would achieve a uniform coverage of the k-space instead of kind of going in a different pattern. So these are the gradient waveforms for my trajectory, and as we can see, they vary smoothly in time. And so because of this, there's not much noise when the gradients are played out in the MRI scanner. So I do this with the use of an integrated development environment for MR applications, and this is offered by Siemens. So this would involve sequence calculation environment for data collection and also an ice simulation environment for image reconstruction. So I've been developing my code in this software, and so now comes the implementation part. So in the implementation part, we would be able to enter the parameters that we want, and we would be able to localize voxels and then do data acquisition, and we would be able to get the data, and which would require further processing. But it's I mean, it's a very good non-Cartesian reconstruction method. So that's why it would have an upper hand over the other other case-based trajectory methods that we have. So this is the simulation of the sequence. This is the RF pulse, and this is the ADC signal data. And the gradients, the gradients are played out, and these are, although these look like triangles, these are sinusoids that vary smoothly with time. And this is the slice selection gradient here. And yeah, that's all about my sequence. And now I'm going to be talking about the future milestones of my project. So right now, data collection has been going on pretty well, but it also requires a bit of code optimization for like data acquisition in phantoms in such a way that the non-Cartesian image reconstruction is a bit easier. And then so this is data reconstruction that's required. And after this, once we are able to map metabolite concentrations in phantoms, it would be followed by trials in humans and eventually patients with mild cognitive impairment or Alzheimer's to detect alterations in brain chemistry and metabolism, which would serve as a very potential biomarker for detecting Alzheimer's, and possibly even it could also be applied to diagnosis of Alzheimer's in patients as early as mid-20s or even early 30s. So that's it. Thank you for listening, and I'm open for any questions that you might have.